Okay, we shall continue with the post-transcriptional modifications, right, by looking at the process of splicing. Okay, so for eukaryotes, right, our genes is a bit different from the prokaryotic genes. Our genes are sectioned into what we call the exons and intron sequences. So within a gene, right, even though we say that a gene codes for a product, but not the entire gene codes for a product. Right? There are segments of the gene called introns that eventually are not part of the mature mRNA, and so therefore they do not get translated. Okay, so the intron sequence do not code for the final functional proteins in a eukaryotic genes. Okay, so after transcription of a gene, right, then obviously the pre-mRNA also contains the sequences that are corresponding to the exons and the introns of the gene. Right, so therefore, the introns, which should not be translated, would need to be removed and the exons to be joined together into a continuous sequence. Right, so this process is known as splicing. Right, so the removal of introns, right, is called excision of introns, right, more chim term, right, and the joining of the exons together is called splicing. Right, so the overall process is called splicing, but then splicing also means just the joining, right? So one word, two meaning. Okay. So how does splicing occurs, right? So in the introns, of course, introns can have all sorts of different sequence. But most of the introns, if not all, always starts with GU and AG, right? So regardless of what is the sequence in between, right? Most, if not all, introns would start with a GU and ends with an AG in the mRNA. Okay, so these are known as splice sites. Okay, so we can recognize an intron, right, with the presence of such splice sites. So what's going to happen is a type of protein called snRNPs will bind to the splice sites. Okay, so if you look at the snRNP more closely, right, it's a complex of protein with a nucleotide sequence within it. Right? So it's a complex of protein and nucleotide and RNA sequence, known as snRNA. So what happened is the snRNA, right, would base pair with the splice sites. Okay, so as you can see here, right, so you have different snRNPs. There are a number of different RNPs. They will come together to bind to the splice sites, and the binding is via the complementary base pairing between the splice site on the mRNA intron and the snRNA within the snRNP. Okay, so once they have bound to the splice site, the different snRNPs would bind together. Right, so you can imagine, right? So they bind to the splice sites, right? The one at the start of the introns and towards the end of the intron. Right, so as they come together to bind and complex together to form the spliceosome, you can imagine that the intron is bent, right? So that those at the start of the intron and those at the end of the intron can come together, right, to form this spliceosome. Right, so this bending, right, this looping of the intron, right, the intron is then looped into a structure called a lariat. Right, lariat here, so they loop some more, right? And you can see, right, the exons, which is the purple color one, are now brought close together, right, as the intron is looped into this lariat. Okay, so the spliceosome would then cut away this lariat, this intron lariat, and join the exons, which are now close together, right? Join the exons together. Okay, so you have the exons joined together and then the intron in the form of a lariat cut away or excised. So that would be the process of splicing. So you can imagine this same process would happen for all the introns in the pre-mRNA. So as a result, right, the mature mRNA would only contain the exon sequence. Okay? So what's the importance, right, of introns? Okay, so introns doesn't really have a function now, right? It's cut away, right? But the fact that there is the presence of introns and that there is a need for splicing then allows for a process called alternative splicing. Okay, So alternative splicing is when, just now you look at the splicing process, it can happen differently, right? So later we look at the process of alternative splicing. But now, as a result of alternative splicing, we can have different mature mRNA sequence from the same gene. And the different mature mRNA sequence can then be translated into slightly different proteins from the same gene, right? So instead of one gene only coding for one protein, right, the allowance of this process of alternative splicing due to the presence of introns allow for one gene to therefore code for more than one proteins. And that means that we can have fewer genes in the genome, which makes the genome shorter, which allows it to be packed into the nucleus, which allows the genome to be protected, and so on. Right? So even though introns itself may not have a function, but its presence allows for alternative splicing, which is important. Right? So now we look at more closely at the process of alternative splicing. All right, so in alternative splicing, right, all the introns are excised. Okay, so remember these are all the introns are excised. Okay, so as long as it's an intron, it will be cut away. All right, because in the past, right, students used to have this misconception that some introns may be not excised and kept. No, right, all introns are removed, but the joining of the exons, right, can be happening in different combinations. Okay, so I'll show you a diagram that will be clearer. Right, so imagine this gene, right, in the DNA, which is transcribed into the mRNA, pre-mRNA, right? So the pre-mRNA has got exons and introns and exons and introns, right? Those not labeled is the introns, right? So as you can see, right, after splicing, right, this is one scenario, right? All the exons, all the 5 exons are joined together and all the introns are excised, right? So this is one form of mature mRNA which can be translated into this protein A. But a second scenario is that there is no exon 3. So this mature mRNA contains a different combination of exons, only exon 1, 2, 4, and 5. No introns. So this mature mRNA can then be translated into a different protein, right? A third scenario, this one don't have exon 4, right? Don't have exon 4, okay? So only exon 1, 2, 3, 5, no introns, right? So this mature mRNA goes on to be translated into a different proteins. Right? So I hope this is clearer, right? So you can see all the introns are excised, as I've mentioned, and exons are joined in different combinations. So some exons are joined, some are not. So that would form different mature mRNA sequences, which goes on to be translated into different proteins. Right, so the exons are, as you can see, exon 1, exon 2 are always joined together, right, in this 3 scenario. So exons 1 and 2 are known as constitutive exons. Then exons that may be joined or may not, right, in different scenarios, in different alternative splicings. Right, these are called the alternative exons. Okay. All right. So just extra info, right? If you look at so far in our research, right, we have found about 17,000 or 18,000 genes. Right. These two human proteome map and proteomics DB, right, are online database of genes and proteins that have been found so far. Okay, so I searched this a few years ago, so the numbers could be different today. But anyway, a few years ago, there were 17,000 or 18,000 genes identified in the human genome. But if you look at the number of proteins, it's much higher. Right? The number of proteins that's identified is much higher. Right? So the only explanation, or don't say only, one of the major reasons, right, for such a high number of proteins compared to genes would be because of alternative splicing. Right? One gene can actually code for more than one protein. Okay? Right, so these two scientists who are still alive today are actually given the Nobel Prize for discovering alternative splicing. Right. Okay, so we have talked about the whole process of post-transcriptional modification or RNA processing. Right, so the the product would be the mature mRNA, which would then be transported into the cytoplasm for translation by the ribosomes. Okay, so we have looked at the whole process, right, from transcription all the way to post-transcriptional modification to produce a mature mRNA. So now as a consolidation, right, can you list down, right, instead of describing all the process, which is super long, right, list down the proteins that are needed for the process of transcription and post-transcriptional modification. So that will serve as a consolidation to what you have learned so far. Right, so you can pause the video, try this out, and then you can resume to check your answer. Right, so firstly, for a eukaryote, right, you need the general transcription factors to bind to the promoter that will recruit the RNA polymerase to form the transcription initiation complex, and then you have transcription, right? So at the end of transcription, right, you have the proteins that cut the pre-mRNA to terminate transcription due to the presence of the polyadenylation signal in the mRNA, and the capping, right, would occur catalyzed by the enzyme mRNA guanine transferase. That's the SNRNPs to form the spliceosome for splicing, and the presence of the polyadenylation signal, right, at the end of the mRNA would recruit the polyadenylation factors, which then recruit the poly-A polymerase for 3' polyadenylation. Right, so these are the different proteins needed to form a mature mRNA.